Good morning and hello everyone. Thank you for joining our webinar today. It's October and Halloween is coming up, so we thought you'd want to know about zombies and cell death. I'm Sally Head-Delquist, President of Immunochemistry Technologies, and yes, I am standing in front of a giant light bright in this picture at the AACR Cancer Research Meeting. And we here at ICT are on a mission to help cure disease, and we do that, ironically, by studying cell death. Cell death is vital for life. So what do zombies, trees, and cancer have in common? It's cell death. And did you know that every day over 50 billion of your cells die and have to be replaced? If they didn't die, you could live 1,000 years. Controlled cell death is so essential to life that all living things, including humans, and even zombies do it too. So cell death is good when it's controlled, like your red blood cells die in 3 to 4 months, but it's bad when it's uncontrolled and lead to cancer or turn you into a zombie. So in this webinar today, I'll tell you about the different types of cell death, like necroptosis, apoptosis, autophagy, pyroptosis, and ferroptosis, and research that's been done to help cure disease, and what you can do to stay healthy, even though we were all part zombie. So I'm Sally Head-Delquist, and I'm President of Immunochemistry Technologies. When people ask me what I do for a living, I used to say, well, I'm a biochemist. I develop tests, like a COVID test or a home pregnancy test, and we sell green and red chemicals to study dead and dying cells. Now I like to say that I'm the Grim Reaper of the biology world. I think that's going to be my Halloween costume this year. I have a BS in genetics and cell biology and an MBA. I started out working in the lab, helping to develop these assays, and now I'm president. And although my initial interest was to understand how things live and grow, I now study how cells die. And Immunochemistry Technologies is a biotechnology company, and we sell reagents to help researchers understand disease. They want to know the biochemical pathway that causes the disease so they can develop a way to fix it. So our customers want to know if the disease or the treatment is killing cells or keeping them alive, and they use our products to do that, to tell them how those cells are dying through what biochemical pathway. Our products essentially help them do an autopsy on the cells. And this research to understand the unique biochemistry of each pathway will ultimately lead to better drugs, and that's why we're all here, to help cure disease and save lives. And I'll show you some ways you can use this research to stay healthy. Assisting me today is Allison Westcott, our Customer Service Manager, so a big thanks to Allison for helping. And to those of you who are out there watching, we really appreciate you taking the time to make this webinar a part of your day. We've muted your phone, but we really encourage you to ask questions. So just type them into the chat box or email them to help@immunochemistry.com, and we'll respond directly to you after the webinar. Also, we're recording this, and we'll post it on our website and YouTube and email a link to you, so you'll be able to watch it again at any time and share it with your colleagues and tell them why they are part zombie too. Did you know that you have 37 trillion cells in your body? And some of them, and by some, I mean 50 billion of them, are dying right now, every day. But that's a good thing. Some of them get worn out and need to be replaced, like your blood cells live for 3 to 4 months, and your bones are always under demolition and reconstruction. You get a new skeleton every 10 years. All of these cells are working together, doing all sorts of different jobs to keep you alive. And 37 trillion cells is such a big number that's really hard to imagine. So think of it this way. If cells were people, and there are 7.8 billion people in the world today, that would be over 4,000 planet Earth. That's how many cells your body has to feed, monitor for disease, and replace. Yes, we are all part zombie, 50 billion cells of zombie. If your cells didn't die, you could live 1,000 years. Each one of your cells has its own time clock that tells it when to grow, when to divide, and when to die. That's controlled by your DNA. And your little DNA has sections on each end of each strand called telomeres that protect the chromosome. The chromosome is this orange X in this picture, and the telomere is the little blue cap at the end. And every time the cell divides, those telomeres get shorter and shorter until they're gone, which triggers the cell to die naturally. This typically occurs after 50 to 70 cell divisions. This is the biochemistry behind aging. Telomeres control our cellular time clock. This only occurs in cells that continually divide, like red blood cells, but not heart muscle. Sometimes An enzyme called telomerase will help repair the DNA, making some cells immortal, which might be happening if you're a zombie. So the actions of telomeres and telomerase may be the cause of some disease, as well as the secret to the fountain of youth. If this process of aging could be eliminated and damage repaired like a zombie, we would live 1,000 years. Now that we know how cell death is hardwired into our DNA, we can see it. We can see it all around us in nature every fall when the leaves fall off the trees. Here's this great cartoon from Mother Goose and Grimm. Attila the cat accusingly asks Grimm the dog, "Say, isn't that the tree you piddled on 2 weeks ago? Now all the leaves have shriveled up and died. Well, Grimm, I hope you're satisfied." And then Grimm walks away ashamed of himself for killing the tree. Well, if Attila were a biochemist, he would know that the leaves were programmed to die and fall off. This is one form of cell death called apoptosis. What? That's apoptosis. It's derived from Greek, meaning leaves falling off. Each year, the cells at the end of the leaf stem naturally die by triggering caspase enzymes and fall off without damaging the rest of the tree. In the spring, the leaves grow back again. Apoptosis is so well conserved throughout life that it is as similar in plants as it is in humans and helps all plants and animals grow and remodel. When a cell is too old or becomes defective, it will self-destruct following a controlled biochemical reaction. This triggers caspase enzymes to dismantle the cell from within without causing damage or inflammation, unlike a cut where there's inflammation and a scab. If the cells didn't die and replaced themselves in a very controlled manner, they would either overgrow and cause cancer or die too soon, causing problems like dementia. Cell death is a vital part of life to keep us healthy. Believe it or not, cell death is good for you when it's controlled, like the leaves falling off the trees, but bad for you when uncontrolled, like cancer cells growing out of control when they should be dying. I'm not really sure about the zombies, but they are scary. And cell death is a vital part of life for all species, from plants to humans, but it has to be controlled. Programmed cell death, like apoptosis, saves your entire body from dying. Without apoptosis, cells overgrow and cause disease like cancer, but with too much apoptosis, cells die early, leading to problems like neurodegeneration and dementia. Cell death is good when it's controlled. During growth, genetic regulation of cell death is vital for embryonic development. The DNA expresses varying degrees of apoptotic factors, such as caspase enzymes, that will help structure the body. For example, some species of salamanders have webbed feet, while others have more distinct toes. The salamanders with the toes have more active caspase enzymes in their developing feet, which remove the webbing between their toes. In humans, there is a high level of apoptosis leading to individual fingers and toes where there was once a solid hand when you were just an embryo. Cell death is bad when uncontrolled. Cell death happens too much or too little, causing disease and birth defects. Cancer cells grow too much, causing tumors, and neurons in the brain might die too soon, causing dementia. It's very important that cell death is controlled. Us, being curious humans, wanted to ask, how does this happen? Why do the leaves fall off and regenerate? How does the body know what to do? And we uncovered many different reasons. Did you know that there are many different types of cell death? Our bodies are really complex and do their best to keep you healthy. Some ways our cells do that is by responding to an accident to form a scab on a cut, triggering the immune response, and even sacrificing themselves to save other cells. Each one of these biological pathways has a unique biochemistry and pathophysiological characteristics, and it's important to understand all the biochemicals involved so we can treat injuries better and develop better drugs with fewer side effects. It's an amazing process that fascinates me. Let's dig into the science behind cell death, and we'll look at an overview of some of these pathways. Cell death is typically categorized into non-programmed or programmed cell death pathways. Programmed cell death is also thought of as regulated cell death, which has an underlying genetic program. And the non-programmed cell death is often called accidental cell death, which is typically necrosis. So necrosis is what we typically think of as a dead cell. It's usually due to an injury, accidental death, or the body's inability to repair itself. That's what we see on the left. The cell simply breaks open, spills its contents into the surrounding tissues, and damages them too, which snowballs and creates more damage if the body doesn't heal itself. Programmed cell death is what happens when the cell gets too old or becomes dysfunctional, and we see that on the right. The cell efficiently dismantles itself and is recycled without damage to the surrounding cells. Scientists used to think that there was just one form of programming called apoptosis, the leaves falling off the tree. But now we know there are several other pathways of programmed cell death that trigger a biochemical response to control the breakdown of old, damaged, or dying cells, like necroptosis and ferroptosis. And that's where it gets really interesting. Necrosis is inherently inflammatory. Your body responds to stop the injury and repair itself. That's the cell on the bottom in the middle with the popped cell. In contrast, programmed cell death can either be non-inflammatory or pro-inflammatory. Apoptosis and autophagy are non-inflammatory, like the natural cell turnover in apoptosis. In the middle left, we see these cells being eaten by other cells, while processes like oncosis and pyroptosis are inflammatory pathways, and they generate an immune response. So like pyroptosis responds to an infection. You see that on the right, like causing a cytokine storm of immune molecules. All these pathways are important for maintaining homeostasis of the body. Let's start with necrosis. Necrosis is a form of cell death that does not involve activation of a programmed cell death pathway to control this breakdown of the cell. It's usually just an injury, but overall, at its most basic level, necrosis is the sum of changes that have occurred in the cells after they've died, regardless of how they got that way, regardless of the pre-lethal process. Necrosis, therefore, refers to the morphological state seen after a cell dies and has reached equilibrium with its surroundings. In turn, a necrotic cell may trigger a regulated pathway, such as necroptosis, because once a cell dies, it still releases its contents, which tends to then trigger other cells to die. It kills them and releases these DAMP, these DAMP molecules, damage-associated molecular patterns, which propagates the inflammation. So necroptosis kind of blurs the line between necrosis and regulated cell death. It may look like necrosis, but it's morphologically similar with swelling and rupture, but it's actually regulated by underlying genetic programs and is regulated by distinct proteins, mainly RIPK1 and 3. Necroptosis forms ion channels that cause the cells to swell and explode, and it's activated by this RIPK1, which requires RIPK3 dependent phosphorylation of the MLKL complex, and the MLKL oligomerization results in plasma membrane rupture, which leads to cell lysis, called the lytic form of cell death. And again, this releases these DAMP molecules, these damage-associated molecular patterns. Both RIPK3 and MLKL are key effectors of injury propagation, and necroptosis represents a form of regulated cell death that's implicated in many diseases, like kidney and lung injury. On the other hand, necroptosis may have a beneficial role by removing infected cells by helping with the modulation of host defense. It may also activate anti-tumor response and help treat cancer. Apoptosis is the most widely studied pathway of regulated cell death and efficiently removes defective cells from the body and remodel it, like the leaves falling off the trees. The apoptotic pathway can be activated in two ways, either intrinsically by intracellular signals from within inside the cell that it's damaged itself, or extrinsically from external signals from outside the cell, like interaction with an immune cell or another damaged cell that triggers apoptosis. And apoptosis is initiated by caspase enzymes, which then cleave cellular substrates to dismantle the cell from within. Caspases are essentially the wrecking ball of your cell. And once activated, the cellular nucleus condenses, the DNA is damaged, and the contents of the cell are neatly packaged into little balls, these apoptotic bodies. And then the cell is phagocytosed, meaning it is eaten and carried away by other cells, and there's not any injury or damage to the surrounding cells, nor is there any inflammation. And on a side note, ICT has many products that can detect caspase enzymes, so researchers can easily track dead and dying cells in vivo, in vitro, and real time, and I'll show you some data at the end of how to look at cell death. So pyroptosis happens in response to an infection or an antimicrobial response to pathogens. It is a highly inflammatory form of programmed cell death, so you get a lot of inflammation. There is cleavage of caspase-1, which is an enzyme, which processes and activates interleukin-1 beta and interleukin-18, which are cytokines, and they're secreted from the cell. There's gasdermin D and E are cleaved, leading to pore formation and swelling of the cell. The DNA fragments, but the nucleus remains intact, which is actually different than apoptosis. And this picture is from a 2020 paper by Yuan Fang from the Nanjing Medical University in China, who now proposed 3 different pathways of pyroptosis. Here you can actually see what I'm talking about. I really like these pictures. This is from a scanning electron microscope taken by Jian Chen and researchers from Xiamen University in Fujian, China. The normal cell is on the top compared with the other dying cells. In necroptosis, you can see the swollen cell about to burst. The pyroptotic cell in the middle is flattening, and it's also forming blebs of pyroptotic bodies. And the plasma membrane will rupture and release the intracellular contents, resulting in inflammation around it. In apoptosis on the right, where the cellular contents are neatly packaged into apoptotic bodies, those are little brown things, the balls, that are ultimately taken up by phagocytic cells in a non-inflammatory manner. And the difference between the necroptosis and pyroptosis is the channel-forming activity of this molecule, MLKL, in necroptosis, which forms these ion channels that lets in water so it osmotically swells, compared to in pyroptosis, they still form pores, but it's created by gasdermin D, which is nonspecific. It doesn't rupture as explosively. So those 2 molecules determine different ways of causing the plasma membrane to rupture in necroptosis versus pyroptosis. Versus in apoptosis, it doesn't rupture at all. It's just naturally phagocytosed and taken up by other cells. Autophagy is another form of very interesting regulated cell death. It's when the cell eats itself. It is the primary system for cleaning the body. This process delivers cytoplasmic or extracellular components to the lysosomes for disposal. And the lysosomes are your internal garbage cans. You can think they help process materials. So autophagy is the housekeeper, and it creates its own internal balls of autophagosomes that fuse with the lysosomes to deliver whatever it is that cell wants to digest. This helps to remove or degrade unnecessary materials like misfolded or aggregated proteins, damaged organelles, cancerous materials, foreign pathogens, viruses, and microbes. And it helps retain and maintain good materials like biochemicals you want, metabolites, and the organelles that are required for survival, function, and development. And here's the biochemistry of autophagy from Rika Kande from Barkatullah University in Bhopal, India from 2019. Senescence is a cellular response that limits the proliferation of aged or damaged cells. It's kind of like suspended animation. The growth is arrested, but the cells don't die right away. Senescence is the biochemistry of aging, and it's triggered by a stressor, such as genomic instability or telomere attrition. That's your telomeres getting shorter and shorter, and that triggers senescence. Senescent cells also secrete hundreds of factors. These little proteins from pro-inflammatory cytokines, again, you can think of the cytokine storm for inflammation reaction, chemokines, which attract or repel other types of cells, growth factors, and proteases, which are other enzymes. All of this action leads to a gradual decline in function of the cells as the cells and tissues progressively deteriorate. There is stem cell exhaustion and chronic inflammation, and there are two main pathways of senescence, both of which repress CDK4 and 6. Yes, deleting senescent cells tend to extend the lifespan and health, but we also found that trying to prevent the telomeres from shortening also increases the risk of cancer. So there are many pathways of regulated cell death. And the last one I want to talk about today is ferroptosis. Ferroptosis is a relatively new pathway of cell death proposed in 2012. Ferroptosis is dependent upon iron, and iron is an important trace element in your body. You have Fe2, it's oxidized to Fe3, and then reduced back to Fe2. Iron is recycled to maintain homeostasis, and any disruption in this biochemistry causes a problem. Typically accumulates lipid-reactive oxygen species, also known as ROS, leading to oxidative cell death. It has obvious shrinking of the mitochondria with increased membrane density and reduction or disappearance of mitochondrial cristae. It does not involve the morphological characteristics of necrosis, like swelling of the cytoplasm and organelles or rupture of the cell membrane, nor of apoptosis. So there's no cell shrinkage or chromatin condensation. There's no formation of apoptotic bodies, and the cytoskeleton doesn't disintegrate right away. And unlike autophagy, it does not form closed bilayer membrane structures like the autophagic vacuoles. And I'd like to thank Ji Li et al. At the Harbin Medical University in China for a nice review paper from February of 2020. And this is from their figure 2. So you can take a look at this pathway. And remember, we're going to post this online, so you'll be able to pause this video and watch it again later. So remember, there are a lot more regulated pathways of cell death, and some of them I didn't talk about today. And this is a really nice table from Mary Chow's paper from 2019 that highlights some of the features and key regulatory molecules involved in some of the cell death pathways. Notice that only 2 of them on this table are genetically regulated. That's necrosis and senescence. The rest have key proteins like caspases or iron or MLKL in necroptosis to help regulate the pathway. Learning about this is interesting, but how does it really help us? You know, research saves lives, and using ICT's apoptosis testing kits and caspase reagents for cell death, our customers have made some really important discoveries. So let's take a look at some of them. What can we do to help our bodies regulate cell death? Well, the easiest thing is to simply eat the rainbow. Eat a wide variety of colorful fruits and vegetables. We've got 37 trillion cells to feed, and some of them are going to go rogue. So destroying these defective cells before they start reproducing is critical to prevent cancer. And our bodies normally detect and destroy these defective and damaged cells effectively through regulated cell death. And this is heavily influenced by our diet, by naturally occurring dietary compounds found in our food. Here are some of the superfoods. These are packed with potent phytochemicals to keep you healthy. Our bodies evolved by eating these plants, and we operate our best when we eat them. They work by reducing inflammation, where you get swelling and edema, and stopping cells from overgrowing. Because I'm a science nerd, I really wanted to know what was in these foods that make them good for us. You've probably heard of some of these, like resveratrol, the red component in red wine that has anti-cancer, and lycopene that's found in tomatoes and helps cells grow. And there's a lot of talk about antioxidants. What is that? Well, it's like rust on a car. You have these extra oxygen molecules, the free radicals, that attack the iron in the metal like they attack the iron in your body. They oxidize it. So if you eat antioxidants, the antioxidant will bind to the free oxygen before it binds to your cells and damages it. Food truly is your first medicine. Can food really help? Well, yes, it can. And there's scientific proof that it makes all the difference in the world. For example, the people of India have the lowest rate of Alzheimer's disease at 1%, while in Finland, it's the highest at 50%. And the biggest difference is the food they eat, especially curcumin compounds. Curcumin and curries are anti-inflammatory, and one of my customers did a really interesting experiment showing that curries may help with Alzheimer's disease. So this data may look simply like red and green dots, but it was a really important experiment. Dr. Milan Fiala of UCLA noticed difference in the diet of Finland and India and showed that in some people, Alzheimer's disease is actually could be considered a blood disorder, and eating curry may reduce its effects. The macrophages are white blood cells that act as garbage collectors in your brain. They normally pick up and clear away the amyloid beta plaques that smother the neurons in the brain. But in some people, the macrophages don't work right because they die young. They die too soon. Instead, these defective macrophages pick up the amyloid beta and they turn green, which is what you see in the top left. But then they die and they turn red, and that's my product is the red stuff. They become apoptotic and they drop the plaque off somewhere else in the brain. These cells don't even finish their garbage route. They just keep shuffling the plaques around from one part of the brain to the next, and it just builds up, leading to Alzheimer's. But Dr. Fiala discovered that curry stimulates these macrophage cells to stay alive long enough to hang on to that amyloid beta plaque and finish that garbage route to get it out of your brain so your body can get rid of it like you're supposed to. So in the bottom row, they pick up the amyloid beta, they turn green, but then on the lower right, they don't turn red, they stay alive. In another experiment done at the VA in Ann Arbor, Michigan, showed that diabetes causes brain damage in addition to other complications. So these are brain slices from a diabetic rat on the left and a healthy rat on the right. You can see there's a lot of yellow in the diabetic rat, meaning that those brain cells are dying. And this occurs in the area of the brain called the periaqueductal gray, which controls your pain sensation. So diabetic patients may have neurodegenerative problems and need additional treatment in addition to peripheral neuropathy. Salmonella is a bacterial pathogen responsible for food poisoning. It may cause gastroenteritis or systemic typhoid fever. And Salmonella thrives in the gut and the gallbladder and causes inflammation. You see it here in this picture as the red dots. When spreading within the host, Salmonella escapes from a cell and enters the cytoplasm to replicate and attack other cells. To escape, Salmonella tricks the cells and triggers cell death via apoptosis by activating caspase-1, and the cell self-destructs, spreading the Salmonella all over. And here you see the red dots spread around. And Salmonella may be controlled by preventing it from spreading within the host. The red Salmonella is shown in the cytocell in and around the infected cell, which is the bright green, which are dying. They're tricked into dying via apoptosis. And one way to stop Salmonella is to stop apoptosis. Studying cell death can help us cure blindness. These are human cornea cells from the eye grown in cell culture. The ones on the left are keratoconus corneal cells, which die leading to blindness. You see they're all green. Versus the picture on the right shows a lack of green staining. Most of them are healthy cells that were treated with a drug. So by studying cell death, we can help develop cures and treat patients before any vision loss occurs. We can breathe easier by reducing inflammation in the airway, by inhibiting caspases, the enzymes that cause cells to die. These are lung tissue sections from mice treated with caspase inhibitors on the top, which shows a remarkable reduction of inflammatory cell infiltration of the airway parenchyma, which is in the arrows below, and also in the surrounding blood vessels of the airway. And you can see a lot of mucus inside those airways. So the untreated mice in the bottom and B show a lot of mucus in the airway. That's the AW, the arrows that is plugging the airway. And you see an extensive infiltration of leukocytes of the lung parenchyma. And the blood vessels in the bottom are pink, filled with eosinophils and other inflammatory cells. So by preventing apoptosis to happen, you can breathe easier. This mucus doesn't happen. This infiltration of the immune system doesn't happen. Remember that research saves lives. I'm really optimistic that we are finding the cures. So even if you're not a bench scientist, you can help too. Support government-funded research, which is more important than ever in this time of COVID-19. Participate in clinical trials, be an organ donor, donate to charities that support medical research, volunteer, and most importantly, take care of yourself and eat curry. 50 billion of our cells are dying every day, and we're all part zombie. So do whatever you can to stay healthy. I hope this overview of cell death was helpful, and we have other webinars that go into a lot more technical detail with each one of these pathways, so watch those if you want to know more. ICT has many fast, easy, and reliable methods that can be used to detect cell viability in functional studies, in vitro, in vivo, and ex vivo, and all of these kits are available online, so check them out, and if you're in the lab bench, you can use them to expand your research toolbox by including our reagents for cell death. And that wraps up our presentation for today. If you have any questions, please feel free to send them through the chat box or email to help@immunochemistry.com, and we'll respond directly to you. Of course, you can always call us. We love hearing how you are using our products and helping you set up your experiments. And again, thanks for tuning in today and for all the research you're doing. And thanks to Allison Westcott, Nick Weninger, and Kristen Pauly, our marketing team, for managing these webinars. We here at ICT are proud to be part of the medical research community and provide research reagents and kits that can help understand and someday cure disease. I'm Sally Head-Dahlquist, President of Immunochemistry Technologies, and it's been a pleasure helping you understand cell death. Have a great day.